Hello everyone, my name is Sasin Taranum and today I'll be talking about mutations, point mutations, and their effect at the protein level. Mutation is a change in the DNA sequence, and point mutation results in substitution for one base for another. There are two types of point mutations, transitions and transversion. Transition, which means mutation occurred from purine to purine or pyrimidine to pyrimidine, and transversion means mutations from purine to pyrimidine or pyrimidine to purine. So how does this mutation affect at the protein level? Let's discuss it over a given DNA template. So the wild-type DNA template that is given here is this, and the mRNA that is transcribed from the wild-type DNA is this. Now we have the proteins from the mRNA that was transcribed from the wild-type DNA is methionine, tryptophan, asparagine, alanine, cysteine, and stop. Now let's discuss about different types of mutations and their effect at the protein level. Let's compare the wild-type DNA with the mutated A-strand DNA. Here we see wild-type DNA has TTG, but the mutated A-strand DNA has TCG. Here G base was substituted by C. Here G is purine and C is pyrimidine, so this is a transversion mutation. TCG is transcribed into UCG, which results in the amino acid serine, but TGG in the wild-type DNA becomes UGG after transcription, and after translation it becomes tryptophan. So tryptophan and serine, these are two completely different amino acids, and this mutation resulted into a different amino acid. So this type of effect is called missense effect. Now let's compare wild-type DNA with mutated B strand of DNA. Here we see TGG, but here we see TGA. So G base was substituted with A. Now G and A both are purines. So this is a transition mutation. TGA becomes UGA after transcription, which is a stop codon. So the changes codon the mutation changes the codon for amino acid that becomes a stop codon. So this effect calls nonsense effect. Let's compare wild-type DNA with mutated C-strand DNA. Here we see AAC in the wild-type DNA, but here we see AAT in the mutated DNA. AAC is transcribed into AAC and becomes the protein amino acid after translation asparagine. Here we see AAT becomes AAU after transcription, and after translation it becomes asparagine. So we can see the both amino acids are same because AAC and AAU both codes for asparagine. This type of effect at the protein level called silent effect. In mutated D strand we see AGA, but in wild type strand we see TGA. Here T is substituted by A base, so it is a transversion mutation because A and T are T is pyrimidine and A is purine. So AGA becomes AGA after transcription and becomes arginine. But here TGA becomes UGA after transcription, which is a stop codon. So this stop codon is changed into a codon that codes for another amino acid, resulting in prolonged translation. So this type of effect is called read-through effect. Now that's it for today. Thank you for watching.